is present in low abundance. It's uniformly Zn negative. It cannot be seen microscopically. It's intracellular. It minimizes immune recognition. It is completely different to infection with M. tuberculosis. It is extraordinarily difficult to isolate and propagate in subculture. It's extraordinarily resistant to lysis. And then there are a number of must-not-dos which govern optimize detection. And just to describe this for you, we developed and published some time ago a couple of pairs of primers for nested PCR. IS900 is now known, has a number of lookalikes in M. avium. 9 is the intestinal ileal mucosa from somebody who does not have Crohn's disease or irritable bowel syndrome. And 2 is from somebody who does have Crohn's disease. And 8 to 3 in ascending order of sequence homology are a representative sample of these IS900 lookalikes. What we see is that beginning here with this isolate from Western Australia, there's a pickup of these elements here, and this is the true one. And this can be distinguished from these by simply sequencing the amplicon, which has the unexpected advantage also to have an SNP in the middle capable of distinguishing between bovine and ovine isolates. This is when Tim will tweak the primers, is we can get an entirely specific reaction, but the advantage of these is, of course, that they're multi-copy. Now, this is a must-do. You take a prepared ribolyzer tube, you put the STS protease K in it, and then you put in a fresh biopsy weighing 20 milligrams. And you leave that for 4 hours, and the biopsy, as you see, dissolves. That releases the map from the tissue, but does not release the DNA from the map. You have to put it in this instrument, jangle it until the tube gets warm for at least a minute. And then, in circumstances where 2, 3, and 7, and 8, the negative controls, and 9, 10, the positive controls, are all reading correctly, you find in this person from Ohio, who had been diagnosed by the Cleveland Clinic with irritable bowel syndrome from 20 years, and was then found to have extensive small bowel Crohn's disease, that 4 in his terminal ileum, 5 in his extending colon, and 6 in his sigmoid colon, the intestine is widely affected with MAP. And this is also something very important, which we found recently, and so has Sally Nasser. And that is the distribution of MAP positive tests in the gut. There's no statistically significant relationship to the distribution of the segments of florid Crohn's disease in the gut, and I'll come to the explanation of that in a moment. Of great importance for the pivotal studies of Sally Nasser and his colleagues, using a buffy coat from blood and culturing the organism in 14 of 18 people with Crohn's disease, and not at all from 15 controls. It's interesting to note that his PCR test here detected with good agreement these, but didn't detect this negative culture and picked up some more. Although larger numbers are needed, it's consistent with the presence of these organisms in a state of dormancy in people who are not diseased, which is not amenable to our current system of culture. And these data have been ably supported by Bentley's work from New Zealand using total white cell from blood, and these were his figures. So as in animals, the disease in humans is systemic. Now, this is the meta-analysis published in The Lancet towards the end of 2007. And there is an odds ratio of 7%, showing that MAP has a highly significant and specific relationship with Crohn's disease, but not ulcerative colitis, although it can infect people with ulcerative colitis. And there are some studies included within this that include really inadequate methodologies. It's a done deal. It should be after a century. Most people with Crohn's disease are infected with MAP in their intestines. And when we do it, we identify MAP infection in the range 85 to 92%. This question here, the prevalence of the normal population, is upwards of 15%, but we can expect to find very variable data influenced by strain of organism and geographical location, amongst other things. Now, this is an absolutely pivotal portion of this story in terms of understanding how this organism is causing disease in humans. And I begin it with a question. How does MAP, which is an organism present in low abundance, which is uniformly Zn negative in phenotype, which is intracellular as far as we know, and which minimizes immune recognition, cause segments of florid inflammation of the gut? And very quickly, MAP infection and Johne's disease in animals is accompanied by chronic enteric neuritis. More work is needed on the specific neuropathological effects of this organism in animals. But here is a ganglion within the enteric nervous plexus showing substantial infiltration with lymphocytes. And the next thing that happens is that MAP-infected animals have a local and systemic immune dysregulation. And I've listed a few examples down here, but the list will go right down through the floor. In humans, immune dysregulation and chronic enteric neuropathy are well known in Crohn's disease. One of the many mechanisms, and there is recent data on this which is very useful, is that mannans released from MAP, but not those from tuberculosis or the types of E. coli that they use, are able to inhibit the killing of E. coli by human monocyte-derived macrophages, indicating that a primary infection by this organism may be responsible for conferring the immune disability. In Crohn's disease, the gross inflammation tends to occur in segments, whereas the observed pathophysiological features of multiple kinds is distributed widely throughout the gut. You've already seen that there's no statistically significant relationship between the distribution of the grossly involved segments and the tests which test positive for MAP. Indeed, we found MAP in the perianal lesions of people with Crohn's disease and in orofacial granulomatosis. So that a model of what is happening here, which is consistent with what we know, is that the MAP infection causes a microscopical inflammation and a primary neural and immune dysregulation. This impairs, as experimental studies have ably shown, the integrity of the mucosa and other critical functions of the intestine. And the segments of gross disease result from the perturbed neuroimmune response to the secondary penetration of microbiota, including educated microbiota, and of course a contribution from food residues from the gut lumen. And it is essential to recognize this two-tier system of disease when we come to consider the very exciting opportunities presented by the advances in genetics. The one study so far that's looked at MAP infection of NOD2 in humans, with a fairly limited number of people, came up negative. There's some interesting data just published from Florida show actually that there is a very highly statistically significant association between NOD2 polymorphisms and the risk of MAP infection in cattle. Now, once again, with MAP, as always, beware. The phenotype of the organism in cattle is wildly different from that in humans. And of course, there is a great deal of work to do before we understand which of the known susceptibility loci may be associated with a primary infection. Now, as a last series of slides, and as a glimmer of hope for people suffering from Crohn's disease, this question is posed: can anti-MAP drugs heal Crohn's disease? In the very early work by Larson at Ames, Iowa, and Deans Rankin at the what's now called the Institute of Animal Health at Compton, Berkshire, showed that these organisms are highly resistant, as are many other MAC, to unmodified Streptomyces antibiotics. But when these man-made chemical modifications of natural Streptomyces antibiotics began to become available in the early 1990s, I and others did a number of pilot studies, and these are the kinds of things we saw. A boy with ileal disease and an intensely hot white cell scan, the disease completely disappearing over this period of time. and yielding a tight white scar in his terminal eyelid that had to be removed. Those are my fingers.